So I'm very honored to have Georgia Purdom as my guest, and thank you for coming here to talk with me.
Pleasure to be here.
So Georgia, tell us about your education and some of your background.
Okay, well, I have a PhD in molecular genetics from The Ohio State University. And after I received my PhD, I then taught at Mount Vernon Nazarene University, which is just north of Columbus, Ohio. Taught there as a biology professor for 6 years. And then 7 years ago, I came to Answers in Genesis. And I have a lot of different roles with Answers in Genesis. I'm a speaker, a writer, a researcher, doing media representative, so doing a lot of different things to really, again, equip the church and help the church to know the truth of God's Word.
So, is it kind of exciting working for Answers in Genesis? It's probably old hat by now. But.
Well, it's never old hat. There's always something new every day, and that's what I really enjoy about my job is the variety. For example, the other day I was listening to a seminar here at the International Conference on Creationism, and I got a text saying, well, you know, this news outlet wants a quote on this particular article from Answers in Genesis, so you need to call them. And so, you know, you never know when things like that are going to come up. I've been able to meet people from all over the world and talk with different people.
Very exciting.
It's a great ministry. It's a great way to be able to use my expertise in biology and in genetics to be able to, again, just help people know that God's Word is true, confirm that, and just spread that word to so many people around the world.
Okay, well, good. Well, let's get started. I want to read a paragraph from our book, my book with Frank Sherwin. This is having to do with the argument of the cell, and so I'll let you respond to this paragraph, okay? The cell is the most detailed and concentrated organizational structure known to humanity. It is a lively microscopic city with factories for making building supplies, packaging centers for transporting the supplies, trucks that move the materials along highways, communication devices, hospitals for repairing injuries, a massive library of information, power stations providing usable energy, garbage removal, walls for protection, and city gates for allowing certain materials to come and others to go from the cell. The cell's complexity, organization, and efficiency is a reflection of either amazing chance and good genetic mistakes or profound intelligence and design. And which of those would you subscribe to?
Well, it's obviously profound intelligence because things like that don't just come about by chance. I mean, you can't imagine, even a city that we have today, like the city we're in, Pittsburgh, there's no way that that's going to come about by just random chance. I mean, people just all of a sudden sort of meeting one another and deciding to do different things. That's just not going to happen by chance. That's purposeful in design. And the same is very true for the cell. It's a very apt description. In fact, as you were saying all those different things, I'm thinking, okay, that's the mitochondria, that's the power.
That's right.
You know, that's the power plant, and you know.
You have the garbage trucks,
that could be a lot of different things, but like vacuoles, and you have little vesicles that do that. When you talk about the transportation, which is personally one of my favorite things in the cell, dealing with the microtubules, and you have these little proteins that literally walk along them and transfer. They're motor proteins, and their job is to transport things.
You see the animated graphics, you'll see that they're carrying this gigantic bag, and what's in the bag but proteins?
Yes.
It's just unbelievable.
It is.
And to think that these things are walking along these scaffolding.
It really is. And one of the things when I do a presentation called Wonder of the Cell, that's one of the things that's always interesting to watch how the audience responds when I show an animation. I show a lot of animations, but the one I always tell them, this is my favorite, and you can debate me on this, but you won't win the debate, because it's just so awesome to see these things just walk along. And really our knowledge of that has only come about maybe in the last 10 years or so that we've been able to really understand, because it almost seems impossible. I mean, it just looks like a little person kind of walking along carrying this big thing on their head, so to speak. And I can watch the audience just sort of gasp, and that is amazing. And they always, when they ask me, what's that one talk you give where that thing does this? And that's like the one thing they remember from it, because it just is so amazing to see that.
Wow. Now, I understand you're doing some work in relation to white blood cells.
Well, what I try to do in The Wonder of the Cell is really help people see the complexity of the cell from the aspect of looking at a white blood cell, and specifically a neutrophil, and just all the things that have to occur for it to leave the bloodstream, go into the tissue, and fight infection. And it is a whole cascade of events, because obviously you only want the white blood cells to leave at certain times in certain places. They can't just leave everywhere. And they're constantly, you know, patrolling the body, looking for danger, looking for problems, and they don't, for example, they don't rush along like the red blood cells. They roll along, and the reason they roll is because of specific interactions with proteins on the surface of the white blood cells and the surface of the blood vessel. And I always call them Velcro proteins, because they form and break pretty easy, but that allows them to roll and not just rush on by. We want them to take their time kind of and check things out. But then how do you get them to actually stop? Because they're rolling all the time. And so the body, again, it's just amazing how we've been designed to do this. And when you think about all the steps that are involved in this, I mean, when I explain this to people, I always say, okay, this is going to take me like 20, 30 minutes to explain this entire thing. And I'm not even giving you all the details. I'm leaving a lot out. Right. I'm giving you the basics, the bare bones. And yet the cell, the body itself will do this in a very short period of time. You know, all these steps have to take place. And you can imagine if any one of these steps was missing, it doesn't happen. I mean, this is the way it has to happen. And so again, to think about going back to, could this have been random chance? There's no way, because you would die until you got, it could not leave until you get that next step, you know, and that next step and that next step. So just the
cell is like this ultimate example of irreducible complexity.
Right.
It's just,
you can't wait millions of years for the next step to happen, because otherwise it can't leave and you die from infection.
And then after it's supposedly created, and then it still has to reproduce.
So it's just amazing how then there's these, you know, when you have a bacterial infection, how the tissue will kind of send like there's a whole bunch of things that are released, a whole lot of chemicals, but some of those go to the surface of the blood vessel, right near that area, and they literally act as kind of like a flag to the white blood cell to say, hey, there's a problem right here, this is where you need to come. And so there's a specific interaction between that protein that's put on the surface and the white blood cell. And then what that does inside the white blood cell is it sets off a whole cascade of events to make changes such that, again, another protein, that's not the stopping protein. I always tell people that's kind of, you know, it's a specific interaction, but it's not enough to make it fully stop. And that's why you have to appreciate how quickly this has to happen, because the protein, it needs to put a certain protein called integrin on its surface in order to integrate literally with the blood vessel and make it fully come to a stop so it knows where to go. But that protein, it has to make that protein first. And so what this initial interaction is to set off a cascade of events inside the cell, so that the cell knows, okay, there's a problem here, I need to make that protein, I need to put it on the surface so I can get in here and I can do the job.
I've got a question that I have no clue as to what the answer is. I'm hoping you can help me. Okay, I sort of understand DNA, and that DNA provides instructions for the assembly of proteins, and those proteins are then used to assemble organelles and have a variety of functions in the cell, messaging and so on. So what about outside of the cell? Our bodies are made up of trillions of cells. Well, there must be some sort of messaging somewhere to say, well, this cell needs to go here and do this, and this cell needs to die so that healing can take place. And where is that inside the DNA is to send out this information? It just seems like
it has a lot to do with the different chemicals and things like that are being released from other cells. I mean, cells are constantly talking to one another, so it's not like they're isolated.
But again, that would be like people talking to each other.
They do it through they do it through chemistry instead of verbal words, like, you know, verbal words like we're using. They're doing it through chemical interactions, and so cells on their surface have receptors, and these receptors allow them. They're essentially their way to communicate, and so chemicals interact with them. And when that interaction occurs, and this is what again is just so mind-boggling, is, you know, there's all these things in the cell, and yet there's these what they call scaffolding proteins, kind of, that are associated with a particular receptor that have these proteins in place that need to basically interact with one another once that chemical binds to that receptor. So it activates this, which activates this, which activates this, so it can get a message to the nucleus, get a message to the cell as to what to do next. And so it is an amazing communication system that I don't even think we've begun. I mean, I studied signal transduction, part of that from my graduate school work, and so it's mind-boggling as to how all of this happened. How do they know, like, when to die, for example,
right.
or you know, and there's all kinds. Some of it's internal, some of it's external. I mean, it's just amazing.
Science is yet to learn so much. Yeah. I'd like to go through several of the organelles in a cell, in a eukaryotic cell, maybe even talk a little bit of the difference between a prokaryotic cell and a eukaryotic cell, and the nucleus.
Okay.
What is so special about the nucleus? Why is that so?
It's the library, you know, it's kind of like the library with all this
the
information.
DNA is in the nucleus.
Right, right. The DNA inside of there. And I always tell people that the nucleus is like an archive of a library.
Help us understand the nuclear membrane, and why is that so sophisticated?
Well, the nuclear membrane is what protects the DNA, essentially. It keeps it inside there because you don't want the DNA to get out into any other part of the cell. As far as we know, and I could be saying something different in 10 years, the DNA is kept safe in there. It can't leave. But as you know, the proteins have to be made outside of the nucleus in the cell itself. So how do we get information from one part to the other part? And that's where what happens is a process called transcription, where you're going to transcribe the DNA into another language,
right.
RNA.
So what we're doing is you're taking a strand of DNA, and then there's a process, a lot of molecular machines happening, and then you have a piece of RNA that's coming out of that reading of the DNA.
And it comes out, and the nuclear membrane has what are called nuclear pores in them, and there are special holes, and they're more than just a hole. I always say research labs could spend their entire life studying one aspect of it, and they wouldn't exhaust it.
Just amazing complexity.
It is. It's
what
amazing.
it allows to come out and go back
because
in.
Because, like I say, because of it having to be protected and kept safe, it can only allow certain things in and certain things out.
Right.
And so it's very important. So the RNA, messenger RNA, because I would say it has a message, and the message is how to make a protein. And so that, so I would say it's kind of like a Xerox. You know, if you were in an archive of a library and you wanted to take something out of the archive, you can't do that. But you might be able to make a copy of it and take it out. And that's exactly what the cell does. It essentially makes a copy
and so it's the RNA
of that
that's
RNA, and that's what goes out, and then the
right.
protein is made from that.
And goes to the ribosome, and from there, proteins are being made. So, so we're just getting a general overview here. Mitochondria. So, again, I am just amazed at just the whole notion of energy. If you were to think of, say, a calculator, for example, you could have, the calculator just simply isn't going to work unless it has not only energy, but the right kind of energy. It has to be converted into a kind that the calculator is able to use. So what is mitochondria? How does that work?
Well, mitochondria are a special organelle. They're kind of like a kidney bean-shaped organelle inside the cell, which is responsible for making ATP, which is our source of energy. So it's going to convert things like all the sugars and things like that into usable forms of energy. Glucose is great, but we need to make it into ATP to be able to utilize it in our body for a lot of the systems. You know.
So it's a usable type of energy that our bodies are able to use, just like the calculator would need the specific kind of battery for that particular kind of.
So it's an amazing organelle because it has this really cool little motor itself called ATPase, and ATPase is what's responsible for making the ATP. And I've seen animations of it, and it just blows you away because you see all the conformational changes that have to occur and how it just literally spins kind of like a motor and puts out that ATP.
And all of those are also made up of proteins, are they not?
Oh, made up of many different parts. Yes. Even when you're talking about the ribosome, that makes the protein, I think in humans it's made up of 82 different proteins. I mean, and that doesn't count other parts.
And of course, the cell would require all kinds of energy in order to produce proteins. Am I right?
Oh yes. I mean, we're talking about those things that walk.
And so here you have this conundrum. If you don't have energy, a converted energy that the cell is able to use, how are you able to create? How would you ever be able to produce proteins?
Yeah, exactly.
Again, requires proteins in order
it's all interlocked. You can't just remove any one component because you realize, it's like the chicken before the egg. You have to have
1,000 times over.
Yeah, 1,000 times over. You've got to have all that in place. Yes, absolutely.
Okay, so we're talking about mitochondria. What else? Oh, the mitochondria has DNA. Why? What does the DNA in mitochondria do?
A lot of the DNA there is specific for proteins that are part of the mitochondria, and that do mitochondrial processes. Some of that comes from the nuclear DNA, but a lot of it is actually in there. As to why it's separate and why it has that, I don't know the answer to that, but a lot of the proteins can be made there and utilized.
I think a lot of people would also be surprised at how many mitochondria there are in a particular cell.
Oh yeah.
It's not like one, well, this is the organelle called mitochondria.
Right.
No, there's like,
especially if you look at cardiac cells and
like
muscle
millions or
cells,
billions
which
in a
require a
particular
lot
cell.
of energy.
I don't know.
Huge
There's
amounts
just
packed in there.
Tons of these mitochondria.
Right.
And this is also an area of great concern medicine-wise, because there's a lot of mutations that are happening in the mitochondria, and it's affecting our age, how well we age, seems to have something to do with mitochondria.
Yeah, and there are, I know of an individual that has a mitochondrial disease, and they suffer from lack of energy a lot of times because, again, it's like you say, it's not just one of these inside every cell, there's a lot. And so you can imagine if you have that throughout your entire body, muscle cells just can't work as well. Especially muscle cells, that's where you tend to get most of the effects. And mitochondrial diseases really vary. Some of them come not because you don't produce a certain protein, but because it doesn't make it to the right location. I always say every protein has an address built into it. A street number, street name, city, state, and zip, so it knows exactly where to go in the cell.
Amazing.
And I see it's not just, people have this idea that things are just kind of free floating around in the cell, but that is anything but there's no truth to that, because we'd have to wait forever for something to get to the right place, and instead things are transported and taken to the right place specifically. So some of these mitochondrial disorders come from just not even being able to get something to the right location. So yeah, it's amazing.
Other organelles. So we should probably briefly say what the difference is between a prokaryotic cell and a eukaryotic cell. Can you describe that?
So yeah, just real simply, a prokaryotic cell doesn't have, it might do a lot of the same things, but it doesn't have them packaged into specific tiny organs, so to speak, inside the cell. It's much simpler than that in that sense. Bacteria are still very complex in their own right,
yes.
But they don't have a nucleus, for example, to encase the DNA. They do have a chromosome, typically a circular chromosome. They do have ribosomes to make the proteins from, but as far as a lot of other types of protein packaging and things like that, they just don't do that like we do in the eukaryotic cells. And we're obviously much more complex organisms, so we have to have more complex, structurally speaking, to be able to accomplish all that our bodies need to.
But because it's an organism, it's a single-celled organism, it replicates very, very quickly, and it is fantastic for scientists for their discovery, for their research. So one of the things that I'll challenge people with, because myself and friends will go to the farmer's market, and we have this display with evolution material and giveaway materials, and we'll talk to students at the university and other people that come by. And so we have these discussions with them about, so I'll mention something like about bacteria. What would it take for bacteria, for example, to evolve into something else? And that whole scenario. But this idea that bacteria are simple is anything but true. And yeah, so what would it take? So I understand that they seem to think that a eukaryotic cell, which is much more complex than a prokaryote bacteria, that it wouldn't necessarily come from bacteria, that it would have had maybe a common ancestor to the bacteria and to archaea cells, and so there's really no single known path of life leading up to these three different.
No, we always say, I never say like we evolved, when people say we didn't, they would never say we evolved from bacteria. They would say we evolved from a common single-celled ancestor of some kind that gave rise to all these. But even when you look at, we're talking about bacteria, even the minimal number of genes that are required for life, I think it's something like, and I could,
it's all hypothetical.
It's all hypothetical, but it's still in the hundreds to thousands of at least, I think it's hundreds if I'm remembering correctly, but that are needed to sustain a very quote unquote simplistic lifestyle. So you can imagine, to get to a human being, all of the information that would have to be added, and you can't just tweak bacterial DNA and get a human being. I mean, they have maybe, you know, like some of them I've studied, maybe a couple million bases, but that's nothing compared to in their DNA compared to a human that has 3 billion bases. And so you've got to add stuff. You've got to add it, and there's no known genetic mechanism that allows that.
So one of the things we talk to them about is kinds. You know, that we believe that God created and He created viruses, He created bacteria, He created these other organisms, and all in their own kind, and that there's variability within the kind, within bacteria. And what we don't see is bacteria evolving into something else. And so I'm not sure if you have anything else to say about that.
There's been experiments that are done, and a really great example of that is Richard Linsky's experiments that he's done. He's an evolutionary microbiologist at I think it's at the University of Michigan, and he's been doing this experiment for over 25 years of culturing E. coli bacteria. And so basically he has strains that have been going for over 25 years now, where he just, you know, they keep putting them in new media and they keep growing. And so what he simulated in the lab is probably millions of years, okay, in the real world, because they wouldn't replicate that fast in the real world. And yet still, what are they? E. coli.
Still bacteria.
They're still E. coli after all that time.
Yes,
so.
I remember one of the students, a geneticist, she was talking about how they've been able to put pressure on certain bacteria to create functions that didn't, they didn't have that function before. So then I asked, but are they still bacteria?
Well, and a lot of times too, it's not with Linsky's work. What he's found is that they did have that function. It just wasn't maybe functioning as well or under those conditions. And so they don't really gain new functions per se. It's usually an alteration of a function they already have to allow that to happen.
Okay, going back to the eukaryotic cell then, what about, say, the Golgi body? Do you know what do they do?
Right, so they're involved a lot in like packaging and processing, kind of along with the endoplasmic reticulum.
Yes.
They kind of work in conjunction, so sometimes things will leave the ER and go to the Golgi. And so I always call them kind of the processing plants. They're putting the finishing touches on things a lot of times, like adding sugars. Anything that goes to the surface of the cell typically has a sugar added to it. They might do some refolding. The endoplasmic reticulum can even be like a quality control area, so if something isn't folded right. Okay,
so let me back up for a little bit. So what you mean by refolding is you're talking about proteins. So a protein is made through the ribosome, and then it goes, oftentimes it's being chaperoned to a chaperonin, which is also made up of protein, and so it gets folded. We don't know how.
Yeah, some of it has to do with its just its linear structure and the hydrophobicity, like whether using a big word here.
You know more than I do, definitely.
But whether the amino acid repels water or likes water, so to speak. So based on some of those chemical interactions, they will fold up into a certain. So some of that is inherent within the amino acid linear structure, such that they fold a certain way.
Okay.
But then sometimes they have to be refolded. Maybe there's something, and again,
we don't know. That's what you're talking about now. Is that so? It may be in a particular structure, and then have to be refolded. Do you think maybe it lost its part of its fold?
I don't know, maybe if it lost it, it's just part of the.
Is it being refolded for another purpose?
Yeah, and we don't exactly know why or how it knows to signal that. It must be something. It has to be inherent in the sequence itself in order for it to know, but somehow it knows it has to be. I mean, there's these cool barrel-like structures where they can go in and get refolded and come back out, and we don't know the magic that happens inside, those barrels. But and sometimes too, like I was hearing earlier in the talk today, they aren't folded right, and can't get it to fold right, and so it somehow either attaches a protein to it or whatever, but it signals it for degradation, and so it gets degraded by proteases. Ubiquitin's involved a lot in that particular. Ubiquitin proteins involved a lot in that, so tagging it to die essentially, because we don't want malformed proteins because they won't do what they're supposed to do.
All right. Another organelle. The lysosome? Did I already mention the lysosome?
Lysosomes are kind of, so they kind of go hand in hand with a lot of those things that we're just talking about, is destruction of things and breaking things down. Even things that might come in from outside the cell, like bacteria, sometimes, or even sometimes proteins, nutrients and things that it takes in. It's got to break those down into usable forms. So those kinds of areas help with that. One
of the things that's just so amazing to think about would be that a cell has its ability to repair. repair sections of the DNA. Is that what we're talking about?
Sure, there are DNA repair mechanisms that are built into the cell, and so if a piece of DNA gets mutated in some way, it can repair it. It just depends on the actual mutation. Obviously, we're repairing a lot, and our bodies are very successful at that. Or we'd be having a lot more cancer. We'd be dead. I mean, it has to be able to do that, because whether it's chemical bombardment or UV bombardment or whatever it is, DNA is getting mutated all the time. But again, we have these specific DNA repair mechanisms to deal with that and to recognize those things.
Now, maybe a similar subject, but off the subject of the cell in a way, is that of mutations. You've done a fair amount of study with mutations, haven't you?
Right.
Okay,
yeah.
so I'm a little bit confused on this topic, because in my book I actually describe why mutations are rare. So I have a whole series of issues as to why they're rare. One reason, for example, is that the mutation has to be in the gamete cell in order to carry on to the next generation.
Right.
So, and of course there's the repair mechanism, and there's a variety of other things. But it seems to me that somebody had contradicted me about that. It's John Sanford. That now we're learning that mutations are happening in us much more rapidly than what we had maybe previously anticipated.
Right, and some of his rates show it could be, hundreds to maybe even like 1,000 mutations, per generation, so to speak, that are new mutations, not ones that we've inherited, but new ones that we've made. And so I think what it does, and just from his studies, it looks like we should all be dead by now. So the fact that we're not extinct,
the human race,
I think really shows the amazing compensatory mechanism, so to speak, or redundancy, whatever you want to call it, that God has built into our genomes so that we can deal with that and not be dead as a result of those things. But we are seeing things like, I think, more disease and more, issues with infertility and things like that. So, I always tell people we're all a bunch of mutants. We have 6,000 years worth of mutations since the fall.
That's right.
So obviously that's continuing to build, and as that continues to build, we continue to have more problems with that. But,
a lot more sickness and disease these days, what we've inherited, than what there was years ago. So even though we may feel that we're fortunate that we have all this modern medicine, that people 2,000 years ago or 4,000 years ago were probably much more better off, because they didn't have all this arthritis and illnesses that we have
Well, and
today.
you've got other organisms mutating too. So you've got things like viruses and bacteria, which are doing their own jobs of, of doing these and affecting us as well. So yeah, all of those things work together on that.
This is so much fun. Thank you so much for coming here and teaching us about the cell and about all of God's ingenuity and His design.
Thank you.
Thank you.